Hello, everyone. My name is Richard Behringer. I'm currently the President-Elect of the Society for Developmental Biology, and it's my great pleasure to introduce Rosalind Candy Lee, whose research contributed to the discovery of microRNAs. So microRNAs are short, single-stranded RNAs, 21 to 23 nucleotides long, that can base pair to complementary sequences in messenger RNAs. And so this is a post-transcriptional mechanism to regulate gene expression that has been conserved during evolution. The 2024 Nobel Prize in Physiology or Medicine was awarded to Victor Ambros and Gary Ruvkin for the discovery of microRNAs. Rosalind, or can I call you Candy?
Yeah, you can call me Candy.
Okay. Candy has been a member of the Ambros lab since 1987. Is that correct?
No, 1985.
Okay. Someone's
Yeah.
got to correct Wikipedia.
Okay.
She is the first co-author of the 1993 paper describing the unusual product of the Lin4 locus in C. elegans, the first described microRNA. So what I find exciting about this discovery is that it was born out of developmental biology studies of cell lineages in the nematode C. elegans. And if we think about the timing, back then, C. elegans was still
a new
niche model organism, not like today. So I've been reading about the discovery of microRNAs before the Nobel Prize announcement because these studies are yet another example of how developmental biology using model organisms have led to important discoveries in biomedical research. So when I heard the Nobel Prize announcement, I immediately recognized your name. So I'm very interested to hear your story and how it relates to this important discovery. So welcome.
Thank you.
It's great to meet you, and thank you for taking the time from your schedule to talk. So I'd like to start off with a few questions about your background. So what was your father's profession?
He was mostly an aeronautical engineer. He went
Wow.
to Northeastern on the GI Bill after World War II, and he, worked in Seattle, Denver, and Wichita, Kansas, which is where me and a bunch of my other siblings were born. So
Kansas.
yes.
And so what was your mother's profession?
Well, we had 6 kids, and so she, worked at home, and then once we started going to school, she started helping out this our pediatric dentist who was just starting his practice. She, became his dental assistant and office manager, and she worked there until into her 70s.
Oh, wow.
So she, was very intelligent and picked up stuff really well.
Mm-hmm.
So,
and did your parents have college degrees or advanced degrees?
yeah, my dad had an engineering degree.
Okay.
So,
okay. And do you have siblings? Well,
there were 6 of us,
okay.
5 girls and 1 boy.
Wow. And where are you?
I am the 3rd girl.
Middle.
Yes.
Okay. And what are their professions?
My oldest sister went to medical school. My second sister was an assistant district attorney.
Oh.
I am me.
Yes.
My brother came after me, and he's a retired lieutenant colonel. He was an environmental engineer.
Wow.
Did wastewater treatment and teaching at the health sciences in Texas and in DC. My next sister is Becky, and she was a chemical engineer at 3M until she retired. She did a lot, she went on many different projects, but she stayed with 3M. And the last one, she started off as a CPA and then decided to go into early childhood education.
Okay.
So that's really been her passion.
I'm hearing science and education.
Yeah, I mean, that was very much expected of us.
Oh, okay.
I think most, Asian families expect, their kids to go to college. Um, you know, I'm 70 years old. And so at that time it was, we'll send our boys to college kind of attitude. So, and back then it was still, you were still able to work your way through college. So actually that's what I did. And it sort of makes me, I own my degree,
Yeah.
I mean, certainly my parents were very proud and very supportive, but they thought, well, I should get a free scholarship. And they didn't think about, well, like everybody at MIT is, smart.
Yeah.
Right. So
So, there's a very prominent female scientist named Rosalind, Rosalind Franklin. So, do you know why your parents chose the name Rosalind for you?
It was just a quirky thing. Who knows what parents are thinking, but my parents named all of us girls with the initials RCL. And somehow they came up with Rosalind. It was Rosemary, Roberta, Rosalind, Rebecca, and Renee. And my brother's named Arthur. So I don't, the only person I ever knew named Rosalind was Rosalind Russell.
Oh, yeah.
Actress. And then when I took German in high school, my German teacher sort of looked at me and said, that name, your name Rosalind, it's so special that Germans are afraid to name their kids that because it puts, too much pressure on them. And I thought, okay.
And so Candy comes from Candace.
Right. That's my middle name.
Okay. So you mentioned at one point you were in Kansas, but where in general did you grow up?
Until I was 9, we were in Kansas, and then we moved for a couple years to Pittsburgh. My father, when we lived in Kansas, he would drive us all the way back to the East Coast every year, with all the kids in a station wagon, so that we could see our extended family. And so he wanted to be closer to the East Coast. And then we finally settled in Philadelphia. So when I was in 7th grade on, it was in Philadelphia.
Which I went to UPenn for my postdoc. Which part of Philadelphia?
Well, I mean, I lived in Springfield, towards Delaware County, near the air, near the airport,
more
southern.
south?
Yeah, south.
I see.
Yeah.
Okay.
Yeah.
And we were chatting a little earlier. You said your parents did not teach you Chinese, the language.
No, because I mean they were actually almost illiterate. They could speak Toisanese quite well. However, they came over as babies. And so, they grew up in Chinatowns, but they didn't need to read Chinese.
You said Toisanese?
Yes, Toisan. So most of the Chinese that emigrated to the US before the 70s came from one city, Taishan. That's a couple hours outside of Guangzhou. And it has its own dialect. And it's quite a large city. We actually had a chance to go back there and visit our village and see my family's, ancestral home, and there were villagers who remembered all my aunts and uncles and my father.
Hmm.
It was really amazing. Um, but everybody came over because of the paper sun. So
What's a
I
paper son?
oh
I don't know.
because you're Filipino, you don't know about the whole
No,
system, but
no.
Chinese were not allowed to emigrate unless they were
Remember that.
relatives. Yes, unless they were relatives. So everybody who was working in the US would go back, and then they would claim that they had all these sons while they were back there. So then
Oh,
those people could come over.
oh.
And so, that's why in Chinatown, when I was growing up, the strength of the family organizations was huge. And
Hmm.
that's because of this whole paper son workaround
Hmm.
to and so that was how my grandfather was able to come to the US, and he built a laundry, and then you know made some money and actually moved the family back to China for a little while
Oh,
until World War II broke out, and then they came back.
do, do you know how your grandfather made the first step to come to America?
No, I
No.
mean, I don't have his papers or anything.
Hmm.
Yeah.
Interesting. All right, I learned something new. Hmm. Okay. Um, so now I'd like to oh, yeah. So when and how did you first show an interest in science?
I was just a really curious person and pretty bold in that was a time when kids were allowed to just free range everywhere without any kind of inhibition. I mean, my parent, people didn't have two cars,
Yeah.
um, and you know, there was no one driving us around, and we would just roam the whole town and think of adventures in our minds and look for Indian arrowheads in the gravel, and just explore everything. And
Hmm.
there it was, I realized, a very, you know, good way to raise your kids, not to be timid.
Hmm.
Of course, we did things that I would be, you know, like when my parents, when we moved to Philadelphia, we moved into this new development, and there was this huge length of sewer pipe that was a drainage ditch through the whole thing. And I used to lead groups of kids through that tube tunnel, and it was just, a great adventure. And you
Uh-huh.
could imagine all sorts of stuff, but like, if my kids did that, I don't know if I'd be happy,
Hmm.
you
Okay.
know.
Which institution did you attend for your undergraduate education, and why did you decide to go there?
I went to MIT, and I got a letter from them after I took the PSATs.
Uh-huh.
And, back then, you know, it was just as it is now, where you get a lot of, postcards and stuff. But,
Hmm.
back then lots of schools would send you these things saying, you will get a full tuition, our school, if you come with that PSAT score.
Wow.
And so that's why my parents were unhappy. But MIT sent me a letter saying, hey, we saw your score and we're interested in you. And so I thought, well, this is like a guarantee.
Hmm.
And my father grew up in Cambridge, Massachusetts. My
Oh,
grandmother
okay.
still lived there.
Oh,
So
wow.
they knew all about MIT, and
Hmm.
so they were quite excited. So I ended up applying only to MIT,
Wow.
except for I applied at that time. Penn State had a 5-year bachelor's MD program that
Oh.
that they were starting for the first time. And
Hmm.
they only took 5 students, and so I didn't, make the cut.
Okay.
So, I went to MIT.
And what was your major and did you have a minor?
I majored in chemistry. I did not have a minor, but I
Yeah.
took a lot of biology courses. MIT is very, very generous in letting people take courses in other other majors, and I took graduate level courses in biology.
Oh,
They
uh,
really have the system where they let you sort of push yourself until you fail.
hmm.
So like, they don't have a unit limit. You can sign up for as many courses as you want, and then
Uh,
mostly like people drop them eventually, or, you know, not in a, blase way, but like if you think you're interested in something, you can just sign up and know that, you know, if it doesn't work out, you can always just drop it.
oh.
But you've tried it, you
Mhm.
know. So I took chemical engineering courses too,
Well,
you know. I mean, I was sort of, you know, flailing around for a uh, you know, uh, what I would do
uh-huh.
after I graduated,
Right.
because, I loved organic chemistry
Hmm.
as a paper puzzle thing.
Yeah.
But then I saw that what you did actually was you stood in front of a hood your whole time, right? And that's what it was back then.
Uh-huh.
You were a single person standing in front of a hood.
Hmm.
And
Were there explosions?
um, I didn't make any explosions. I mean, there was lots of fun, MIT hacks where, people would create explosions in the dorm and stuff like that.
Oh,
So
okay. Um, so what year did you receive your degree?
in 76.
76. Okay. And then when you were an undergrad, did you pursue any research training opportunities outside of your classes?
Well, one summer I was, MIT has a extensive undergraduate research opportunities
Uh-huh.
office. Um, and I uh, had a job involving research with cyclic AMP, which was like the hottest molecule back.
Mhm.
Everybody was cyclic AMP.
Uh-huh.
And so the professor said, well, this is what I want to do. Why don't you try and do this and measure cyclic AMP levels? And there was actually a group of 3 of us, and none of us could get this assay to work.
Okay.
And we spent the whole summer
Uh-huh.
going, because you had to go to the library and look through the literature and try to find some kind of alternate assay
Yeah.
to make this thing work. And
And
then
there's no PubMed back then.
yeah, there was no PubMed. And yeah, so I spent a lot of time in the library. And then we finally came to the conclusion that people were not getting uh, this was not an optimized assay by any means,
Hmm.
and it was not going to be accurate if we performed it.
Oh,
But that was the standard.
yeah.
So that just sort of like, oh my God, because I thought if you publish something, it's real. That's this,
Mhm.
and it sort of burst my bubble
Oh,
about research.
oh,
So
okay. All right. So you get your degree in 76. Because now I'd like to switch to the research.
mhm.
Um, and so for context, there was a mutant worm described in the 1980 paper by Horvitz and Sulston and published in Genetics. It was called E912, which
Mhm.
you're very familiar with. So apparently the E912 mutant was isolated by someone named P. Babu using P32 decay. Um, it's a very minor little thing in that paper. It's just, oh, we got this mutant. Um, so I'm curious, what does the E912 mutant worm look like?
It's very long and thin, and it doesn't have it never makes an adult cuticle. It just keeps on reiterating larval stage.
Oh,
And so then it can't lay eggs.
Okay.
And so then what happens is the insides develop to adulthood, but the
Yep.
cuticle does not.
Uh-huh.
And so it would keep reiterating larval stage, except that then the eggs start just piling up insides because you're not laying them. And then eventually the eggs hatch inside the mother.
Okay.
And that's the end.
Okay. So my C. elegans friends say that the worm bagged. Is that
Yes.
bag of worms type phenotype?
Yeah, I mean, the larvae, when they hatch, they just basically eat the mother from the inside out.
Okay. All right. So that's a dramatic phenotype.
Yes.
Um,
So that was, someone automatically would
yeah.
notice. Yeah.
And so if I understand correctly, this is about cell lineages being altered or reiterated and not proceeding like they should. Um, so with this kind of brief context, could you tell us about the research and experiments that led to the 93 paper and the discovery of microRNAs? Because you're starting off with a funny worm and cell lineages was great, fundamental. Um, it led you to something quite incredible that maybe,
Just by happenstance,
yeah.
right? So just by, Victor being really interested in this worm. And, we thought it would be straightforward. You'd, walk to the area, and then try to clone it and sequence it and figure out what it did. That's the usual thing. And it turned out, because this was generated by radioactivity, that causes huge, deletions and rearrangements. And
Mm,
there was only that one allele of that
yes. Uh-huh.
gene.
Uh-huh.
And so, um, Victor was, very curious about this because he knew from watching the cells in the Lin4 divide what happened, what was wrong. And
Mhm.
he got into, heterochronic,
Mhm.
that's the term he was using. And so he really wanted to learn what controlled this,
Yeah.
what was the timer that was doing this. And
Actually, what was the year that you started working on this yourself?
um it was like, 87-ish.
87. So I do have to ask, graduated in 76, start working as a C. elegans molecular biology and whatever in
Mhm.
87. There's 11 years. What happened in between?
So I just worked while Victor was in graduate school. Um, and I would
Worked
work in
in
a lab for a
chemistry?
while.
Oh, okay.
No. I worked in first cardio, um, cardiology lab, studying, atherosclerosis. Then I went and studied yeast cell cycle.
Okay.
Then I worked on a SARC project.
A what?
And SARC,
Oh,
the
the
um,
proto-oncogene.
virus. Yeah. And so I really was given a lot of independence
Mm,
and encouragement by the PI there, Lambo Chen. And, it was because he worked with Victor when Victor was an undergraduate. And
mm,
he just thought Victor was amazing.
mhm,
So when I said, this yeast stuff is just not for me, he said, well, I have the thing opening and I'll treat you just like a graduate student.
mhm.
So, I mean, that was really, really great. And then I went to work for David Baltimore
Oh,
and I worked with different postdocs and graduate students. And then um, he needed somebody to sequence Abelson virus. And so that's what I did for the last couple years there. And then I decided, I had a kid. We had our first kid during that time, and
I guess we should back up and just say that you and Victor are married.
yes, yes, yeah. And so
Now there's a kid.
now there's a kid. And so, I needed to make some more money. And I went and worked at a biotech for a short time
Hmm.
and had my second child.
Oh.
But then I realized, like, biotech is no it's actually more you're under more control of, from the above, about what you do, how you do it, and if you don't accomplish and get the result they want
Mm.
in a certain time,
Yeah.
then they're mad. And it's like, well, that's not how science works,
Yeah.
right?
Uh-huh.
And so after my second kid, Victor's had started his lab the year before,
Mm.
and his first technician went off to grad school.
Uh-huh.
And so I said, I could, go and work for Victor for a while and just see, pick up some different skills, um, and do that. So,
And the rest is history.
yep.
Okay, so tell us about this worm and the discovery.
Well, I mean, you have to put this discovery in the context. This was 30 years ago.
Yeah. Uh-huh.
We didn't have, genome sequencing. We didn't
Mhm.
have any, you know, we didn't even have uh, computer tools to align stuff. So at that time, Alan Coulson and John Sulston were creating cosmid libraries to cover the whole worm genome,
Mhm.
but there was no cosmid to cover Lin4, so
Mhm.
where they mapped it genetically
Mhm.
in the chromosome. And because it was the only allele, we had nothing to try to
Yeah.
grab a hold. So I had to make yeast artificial chromosomes
Well.
and use pulse field gel
Mhm.
electrophoresis, which was
Yes.
a brand new thing
Yeah.
that had just been invented. And that allowed me to, you know, probe with yaks and try and narrow down where, the area in the genome where Lin4 was. And then I just kept on, cutting away, I mean, people don't use restriction digest anymore, even. I just like, it blows my mind, but you do 10 different restriction enzymes and try to see, where it is and see if you can,
Mm.
divvy it up. And so I just kept doing that. And then, Rhonda came into the lab
Mhm.
and
And Rhonda's last name is?
is Feinbaum. So
Rhonda.
she started as a postdoc.
Uh-huh.
And once I got it down to about 800 bases, I gave it to her and she injected it into the mutant
Can I
rescue.
ask a technical question? I guess first of all, when it's down to 800 bases, are you surprised?
No, I mean,
Okay.
800 would be a pretty good protein,
Okay.
you
And then
know?
as far as injections, so this is, I guess, transgene rescue.
Hmm.
When was transgenics developed in C. elegans?
Well, that was at the time, Victor was uh had started his lab at the same time as this other professor, Dan Stinchcomb.
Uh-huh.
And Stinchcomb actually was, you know, uh as he was a postdoc with Hirsch, and he uh started working out what it takes to inject,
Okay.
right? And so that kind of technology was shared in the lab. And then, Craig Mello,
Mm-hmm.
Dan brought Craig Mello with him
Mm-hmm.
from UC Boulder. And so actually Craig did an over expression experiment that we were just, blew our minds.
Hmm.
He injected like, just a couple worms, and then he got a phenotype that was like the overexpression of Lin4.
Hmm.
And so we were just like, everybody, just keep your mouth shut because, and then Craig went on to inject 500 worms that weekend
Wow.
because he was so excited, and he never repeated it. So, that's the kind of thing like he really, um, so I mean, 800 basis
Okay.
is a, and so I sequenced it, and I was like, there's no start site, there's no
Hmm.
anything in here,
Hmm.
you
Hmm.
know, and we just couldn't figure it out. Um, and there's no open reading frames. I tried like, you know, all the different frames and seeing if, I missed a base on the
Uh-huh,
sequencing. It
uh-huh,
didn't make any sense. So then Victor
uh-huh.
said, we got these other, you know, a species,
Hmm.
and we can just see if they have it. So I made libraries from those species and blotted using, the Lin4 from C. elegans against it. And sure enough, there was enough homology that and so then I cloned those. And then
Okay.
once we had those,
Uh-huh.
we could do this alignment.
Yes.
And the only parts that aligned, and I mean, it was amazing that they aligned, perfectly.
Oh,
And, so we were able to get it down pretty quickly. And then we said, well, this has to be an RNA. I mean, it's just,
you make that sound
there
so
was
easy.
there's no open reading frame. I
Okay.
mean, like there may be a 3 amino acid protein,
Oh,
maybe,
no, no.
with no start or stop site,
Uh-huh, uh-huh.
So you know, um, we started doing northerns, and, saw, that there was a band, obviously in the wild type that wasn't in the mutant. So we knew that was it. And so,
Does the mutant have a deletion of that?
yes, it has a huge deletion.
Oh.
So we actually haven't mapped uh, the absolute ends
Mhm.
of the E912.
Oh.
Um, because we were sort of focusing on doing the cloning, and then
Right.
Rhonda did some mutagenesis and isolated another Lin4 mutant that was a point mutation, and it was a
Okay.
point mutation in the sequence
Oh,
that we were interested in. So we
okay.
knew that that had to be,
Hmm.
so
Okay. And then you guys are kind of doing this in parallel with the Ruvkin lab, sort of?
yeah. So when Victor and Gary left the lab, the Horvitz lab, at that time, the worm community was so small. Basically, people said, well, I'll take this part and you can
Hmm.
take that part,
Yeah.
and we won't step on each other's toes, and we'll just
Mm.
work our butts off on our part and see
Uh-huh.
if, it, you know, um, congeals. And so when they each started their labs, you know, Gary took the, Lin14, and Victor took Lin4, and I guess Lin28 too.
Mm.
Um, and so once we knew what this, yeah, because we knew that it had to act in the Lin14 UTR, right? Because if you take away the UTR,
Hmm.
the control goes away.
Mm.
It's the same phenotype
Hmm.
as a Lin a Lin4 mutant. So Gary then sequenced from all the Caenorhabditis strains too, and then he aligned his,
Hmm.
and then saw that there were these exact conserved areas,
Mm.
and
Do
that's
you
what.
remember when that aha moment happened?
I
There's a,
think it was like in, around, the summer of
uh,
1991, maybe.
This is a phone call or something like that,
Yeah,
exchanging data.
yeah.
Hmm. For a while, I, from what I've read, for many years, people thought this microRNA Lin4, Lin14 was some unique, only C. elegans limited phenomenon. Um, but then, can you tell us about your research and experiments that resulted in your 2001 paper, where you showed that it's not that, it's actually evolutionarily conserved?
Well, we had been looking for a long time for another microRNA,
Uh-huh.
right? Just even another one in C.
Yeah.
elegans. And
Yeah.
then, um, you know, Gary Ruvkin's lab showed that LET7, which is
Hmm.
a lethal gene, was a microRNA and it was conserved in
Hmm.
humans
Hmm.
completely.
Yeah.
And so then that said, microRNAs are real. It's a universal control mechanism,
Uh-huh.
and that there have to be many more, right? There's not just these two. And so, we tried all sorts of convoluted ways to do it, but we didn't have the tools.
Hmm.
And so this was during, the time when they were sequencing, they had sequenced C. elegans, and now they were sequencing some of the other Caenorhabditis species.
Hmm.
And so what we wanted to do is find areas that were conserved and had made hairpin structures and
Hmm.
or possible uh you know microRNAs. And so I sort of went at it the back way. In Victor would identify areas where these microRNAs might be, and I'd order the mutants to see whether I could see, and we didn't find anything that way.
Hmm.
But then he, Tom Tuschl developed this way of cloning small RNAs, and that was the breakthrough.
Hmm.
And so when Victor heard that, then he started making libraries, and then we were able to fish out, a number of small RNAs. And, we were just thinking like nobody will think about this except us, right? We're all on Happyville. And then Victor got a request to review two papers, one
Hmm.
by Tom Tuschl and one by Dave Bartel.
Hmm.
And, Tom was doing it in flies, and Dave Bartel was doing it in C. elegans. And so
Hmm.
Victor said, why, I have a conflict of interest,
Yeah.
and so, but then he said, we've just got to get this paper out, you know, and so we had not, we hadn't made any figures. We just had, raw data. We hadn't scanned the gels and made pictures or
Hmm.
nothing. And so Victor basically, they gave him 3 days to submit the paper. Victor goes, oh, we got a paper. And, you know, uh, and he tried to, get a, competing journal
Hmm.
interested. And they said, they ask for the moon and the stars. You have to tell us the
Hmm.
mechanism, the blah, blah, blah. Yeah. And it's like, you know, and so he went back to science and said, we have another paper that's a companion. Would you publish it
Hmm.
alongside and have them consider it alongside? And he said, well, if you submit it in 3 days. So Victor stayed up and he created a standing desk for himself with a styrofoam box. And so he would not fall asleep. Uh, Fortunately, one of our boys, um, was old enough that he could. I think, yeah, he was driving, and so I just said, here's our credit card. Make sure your brothers are fed, and he did, and they're still alive. And I spent the whole time like just running gels, making northerns, blotting, confirming, um, uh, you know, making probes, and then just making figures. So that was, I never thought I'd be pulling an all-nighter at that age, but it was, it was exhausting. So fortunately, science agreed, and then the paper was in such a rough state that the reviewer said, well, yes, this is important, but the writing is horrible. There's so many typos, and the the figure legends don't match up, with the lanes, and so so we had time to, fix all that in the review process. So that was great.
Do you think you still have it in you to do another?
No. No way.
Okay. So this research was born from developmental biology studies, identified a fundamental mechanism that regulates gene expression. What is the current thinking, or are you guys thinking about how to exploit this discovery for therapeutic purposes?
Yeah, I mean, it's, we're lucky because at UMass, we have this incredible, chemist on the faculty, and she's been doing, RNA therapeutics against, neurological diseases, and, knocking down genes, like, the ones that have the, the small repeats, all those, all those neurological diseases. And, she's having a lot of success. And we recently learned of a group of patients that have mutations in their Argonaute genes that, are the proteins that accompany the microRNAs to their, and these are hypomorphs. So they poison the whole system. And, apparently they, grab a hold of the microRNAs and just remove them from it, and don't release them. So the other Argonauts can't step in and take over. I mean, if that Argonaut was just gone, that would be fine, because the other ones would compensate. But because this one, Argonaut has, and now there are a couple other mutations in other of the Argonaute family, other Argonaute human family. And it causes, a lot of developmental delay. The kids seem, we went to a meeting, and it was just amazing because a mother whose child has this Argonaute syndrome is a scientist. And so she made it her mission to create an alliance of the patients and the families. And, she has a Facebook group. It's called the Argonaute Alliance. And, they share their mutations with the scientists. And because they are so open and giving, these mutations, we went to this meeting in August, and already these labs had done cryo EM structures, and modeled the mutations. And Victor sort of got a head start in that he had seen the doctor who was helping these patients, Amelia Piton, and she told him what the mutations are, and we modeled them in our C. elegans. And then our graduate student, figured, he did, all these experiments to, figure out the, expression patterns in each mutant and how they changed. And so it's just an incredible, for a basic research scientist to be able to interact with patients, and basically we're handed, this mutation is important. Mutate this amino acid. This is important. And you know, if we can, I mean, this obviously helps us understand Argonaute and how Argonaute's working and how, what the perturbations and what, the important structure is. And so, you feel a little bit weird. These are people with real
Right.
disabilities,
Exactly.
but they are so incredibly generous
Mm-hmm.
to share it with us. And if we can, knock down these mutant Argonauts,
Mm-hmm.
they seem to have a lot of plasticity in their brains. So they
Mm.
are delayed, but they
Yep.
slowly, like, maybe they'll walk at 5 years old.
Mm-hmm.
Then they'll talk at 7 years old.
Mm-hmm.
It's very much delayed, but they make progress.
Okay.
And so, I know, I mean, it's very hard to talk to the parents, and they're just looking at you like you're gonna do
Mm-hmm.
something and help my child. And
Mm-hmm.
that's really a incredible motivation,
Mm-hmm. Mm-hmm.
when you talk to people. So that was a very emotional meeting.
Well, this has been wonderful. And thank you for joining me today and tell your story of your scientific discovery. Thank you.